I know. Find out. Is that you, Heather? Or Liam?
It is Heather.
Oh, cool. I gotta turn up my volume because I can't hear it. It's too bubbly in here. Can we? Yeah. It might be easy better. I'm not like. Okay. Mm. We know you know how to do this, Heather. If no one else comes, we might just call it. But we'll see.
Okay. I think everybody's just sick. Like everywhere you go, people are just hacking and coughing, and it's like I have friends in different school districts and they're all reporting the same thing.
I've got a thing going on today and I'm not feeling 100% either. So I wonder if I picked that up.
Yeah, I know. I got yesterday. I was like sneezing all day, and then it was like the school
Yeah.
tissues are so awful. I was like, my nose by the time I went home yesterday was like
Oh.
bright Santa Claus red. And so I brought in my own tissues today because I'm like, I can't take it. I was like, I have this Devil's Club salve like all over my nose. So I'm like, I don't even care what the kids think.
Yeah,
I
I
was
was afraid to test for COVID. I was like, oh.
I know some people on Kodiak that were testing positive, like, and that seems to be like what's been happening, but
Max,
they were very sick.
what's the scoop with bugs on Kodiak?
There's definitely some stuff going around for the stomach
Yeah.
bugs and yeah, a little COVID.
Mm.
Yeah.
Oh,
I just haven't been going out or seeing anybody. So I'm healthy.
yeah.
But
Yeah.
I just picture you like in your little apartment with your hydroponic.
exactly. I got them right here.
Oh, cool. Hey,
Little bucket.
how did the biologic things end up? Did they end up, you said you figured out some stuff with it, or did it end up working for some of the farmers?
Yeah. I just had to cut some of the parts and make the seals better, and
Oh,
lower them a
good,
little bit. But it
good.
seems to be good. And the other thing that they have is there are these pumps called aero mixers. They're
Mm-hmm.
pretty strong, but all the farms have at least one or two of those. And that could be another good pump that maybe I didn't know if it's too strong. But it's
Oh,
just a good mixing pump. Um, good for
well,
aerating water.
yeah, getting that aeration going is kind of important.
Are you worried about too much movement or aeration or no? More the lack?
No, I don't
No.
think.
Okay.
Yeah, there can never be too much movement, right? Unless it's spilling out over.
In the teeth?
Yeah.
Yeah, the only thing that, no, I don't think so.
Okay.
The only thing, it's gonna knock off nematodes faster. It's gonna, they're just not, they don't deal with disruptive situation anyway.
Yeah.
But, no, I wouldn't think so.
Okay. Yeah, I didn't know. So some of the ones that are more delicate, fragile might not survive,
Oh,
but
hey, Kelly. Yay. Oh, good to see you. So there's a teacher from down in Anchorage, and I hope our decision to do this today brought you on to check it
Oh,
out.
really? Yeah, so we've been brewing the mimetic onotype for a couple of weeks now, so it's been fun. We're doing a little experiment with it, trying like side by side with different things to see how the plants grow. So I'm excited to see hear from Scott.
Cool. Do you hear that, Scott?
Yeah, great.
Yeah, cool. All right, I'm gonna move my camera so I can bring Scott into the mix here. I'm just gonna move my computer so we can get it set up for both of us to see. What do you think? We can move it back.
But you can see what I'm doing. Here
Yeah.
I am.
Okay, we're
Here's
gonna
what I'm doing.
we're gonna put it up a little higher.
You can kind of see it.
Yeah.
So this is our group. So we have, who all do we have with us? So Bogardus, Heather Bogardus.
Yeah.
And who, I know her, but I don't know the other two. Who are we have?
Okay, do you want to introduce yourself to Scott?
Sure. Max Lyons calling in from Kodiak. Working for small nonprofit here on the island with the farms in the region.
Great.
These Luke pick
Great. And
around.
you were able to go to the session that Mel and
He
Liam.
organized it.
Okay, great. Awesome.
Yeah.
Thank you so
He's
much.
the man that
Of course.
organized it.
Yeah.
I'm going to give it, yeah, and who else? Okay, well, nice, thank you. And then I'll introduce myself.
And you got Kelly. Do you want to introduce yourself to Scott? Kelly gave a great presentation.
Yeah.
On what he's doing there.
So we met briefly on the side of the river at some sort of a food
Yeah.
place in Fairbanks.
Yeah, you look familiar.
And I do the hydroponic program and farming program at King Tech in Anchorage.
Okay, sure.
So we
Sure.
got high schoolers. We're doing, like, right now we're smoking salmon. We're doing
Mhm.
orchard work, garden work, and hydroponics. So kind of covering a lot of ground.
Nice. Oh, great.
Thank
Well,
you.
I think probably you guys know who I am. I'm Mel's co-worker here at Fairbanks Soil and Water, and I do agriculture and natural resources education. But one of the things I'm going to give the much more layman's version of mimetic one-tie, very layman's, because I'm actually putting together a workshop right now, so I want to be able to speak this way so that everybody can get this and utilize this. So any gardener can use this. You don't have to go into the science too deep to understand how it goes together. So what I'm actually going to do is prepare. I have my if you could just angle. Okay, you saw my ugly mug now. Now you get to see the other things.
But I was thinking put it up a little bit higher.
Okay.
What
Yeah,
do you
whatever.
think?
Yeah, she's going to adjust the camera.
I'm gonna adjust
So
the camera
have
so you
a-
can see a little better.
What I do have here is a 15-gallon conical induction tank. It's used in the ag industry. I'm holding it up here so you can see it. It has 3 legs on it. They were very easy to get a hold of until the last couple of years, and they've been very difficult to get a hold of since all the recent disruption and everything. They're made in the Midwest, and they would normally sit on a agriculture sprayer tender, and that's where they introduce the chemicals into the big bulk batch. So the chemicals go into this tank gets flushed out. But what we found it works really well for is a bioreactor, and it keeps everything in suspension. That's the really big benefit of it. Unfortunately, here in Alaska, we don't have these super available to us, where as you can simply use a 5-gallon bucket all the way up to a 55-gallon drum. As long as we just need movement. Air stones work in the bottom, but the induction tank definitely is the ideal, but it's not always available to us. So we work with what we have. So I found like a 30-gallon drum worked really well at home, and I had one of the flat, and of course my phone's gonna ring now.
Whoops. Scott's gotta go turn off his phone. I'll be right back.
Okay, sorry. Yeah, that rings in my ears too.
Oh, no.
So any kind of, and I know you guys have heard this spiel before. I'm practicing doing my layman's version of this. So what we're trying to do is several things. We're trying to reduce the amount of salt-based fertilizers going into either soil-based or hydroponic systems. So this is a universal system that works both ways. It's a system that can be applied to soil directly into the hydroponics nutrient solution, or it can be a foliar solution that is sprayed on. I like to look at the foliar solution is a much more immediate. It's like putting in an intermediate, like putting in a PIC line into the plant, gets the items in there quicker, whereas the in the soil or in the water interacts with the root system a lot more as I feel like it properly should. So I'm going to I never fill my container to capacity because of the foaming and the off gassing that goes on in the process. So I'm gonna put 10 gallons of water, and I've already blown down water, so if we need to be careful to have fresh water, which means we don't have any salinity in it. And then we also want to make sure if we're pulling from a city system that it doesn't have any chlorine in it, because that is antimicrobial. And we'll talk about some other antimicrobial things that we don't want to put in that we don't naturally think of as having that nature to it. So we keep a 35, 40 gallon trash can back here. That Mel and I use for our water resource. So we put our city water in it, and it's constantly has stones in it and is blowing down all the time.
The air tone.
So I'm putting 10 gallons of this water that's blown down into my induction tank.
And you can use a garbage can for this
Yep.
too, because we've done that at the school.
Trash can, 5 gallon bucket works. My rule so most teams, the school of thought is that you mix a batch, you brew it for a couple days, and you use it, done, and you start your next batch when you want to use your next tea. There are arguments, but I agree with a lot of the arguments. So I keep mine as a perpetual system. What happens is this is the survival of the fittest in it. And smaller things will go away. We're going to use actual soil today. So we have protozoa, nematodes, bacteria, and fungi present in it. Our bacteria is going to thrive the best. Fungi is going to do well, but we'll ultimately lose our protozoa because it won't reproduce in this system, and nematodes will get killed off just because it's too violent for it with the airflow going through. So how do we keep that perpetual? Well, let's go ahead and build it first, and then we'll talk about that. So what I like to do first, which I didn't get out, is I like to grow my own. I like to know everything that's going into my system. So even the plant material I'm going to use to feed the biology that's in the soil is a known source. So I typically grow in our nursery. I'll have clover growing in there, or I grow chickweed just to keep everybody entertained. Those are great resources for this. I don't like to pull like chickweed out of our dog yard. It may have something, I don't even, that's arguable whether that would be a safe resource or not. So I grow my own hydroponically, basically growing microgreens, and then harvest those. And I typically either use them right away or I freeze them. So today we're going to pull on our resource of the freezer. And so when Mel and I harvest out of our hydroponic systems here in the office, if the crop doesn't look beautiful, typically I'll freeze it for this resource. Or if we have an overgrowth, we typically give our lettuce to the soup kitchen or to the food bank locally. So you have nice miniature romaine heads that I have here. So 10 gallons of water in this. This is not an exact science. I think Liam put together some more specific numbers to guide people. But for class demonstrations, if we're going to be in a classroom for a week, we like to load it a little heavy so the microbial action just goes absolutely haywire and the kids can see it right away. What you're going to see right away is I'm going to introduce the ground lettuce into the tank. And we're going to see a lot of off-gassing immediately. That's because of the bacteria that exists on the leaves of the plants. And you're going to see that start to activate. Then I'll introduce the soil to it. In the next day or two, we'll see a different phase of off-gassing go once all that biology starts to activate in the system. And, it's like a bunch of rowdy teenagers. They like to do they like to eat and they like to multiply. So that's what's the big party's going on, and it is kind of a survival, and we end up with just a few things left. So I like to enhance that every 2 weeks. I like to bump it a little, not with the same quantities I'm putting in today, but feed it with a little new diversity in the introducing some of that diversity of microbial life back into it in a couple of weeks, as well as feeding it a little bit more, but only like a tenth of what we're going to feed it today. And I also, it's like a sourdough starter. In the fact that like when mom kept that in the refrigerator, she would only say, okay, we use this much and we have to stop here. We can't take it below this line so we can keep all of our life in it. And when we feed it, it has something to grow with. So I like to use when I grind, oops. Oh, I was pulling you with this. I guess I have to run it there. I use a Ninja system. I don't use a regular blender. Mel has regular blenders that I would love to use, but the problem is with viney things and leafy things, it tends to just really wrap around the base of the blender. And the Ninja has this cool little system, which is novel, but actually works quite well. And it has the blades up the shaft that do the blending in it. Works really well, even when I use really stringy things or really viney stuff as my source. I'll try to work, doesn't work together well. So
I almost came and got it to do the paper maker thing.
I use a lot of water and very little vegetation. The kids that when I have students do this, it just works really well. So about like 95% water. And I'm probably going to grind. Well, my heads start to fall apart in here. So, maybe I'll use like a gallon or two of volume of lettuce. I may not grind up that much here just to show you guys, but so I'll put 2 or 3 heads of the romaine that I grew.
It's okay if it stays frozen, it doesn't matter.
Yep.
Doesn't have to
That
be.
bacteria is good. We just don't want it to get hot. That's our bacterial enemy. One of them. It packs on in. Kids love this if you're doing this in the classroom. I don't know how many, I know Heather's an educator, the other 2 educators as well.
Well,
I don't know, maybe.
Kelly is for sure.
Okay, we have, yeah,
and
that's right, at King.
Max has been doing something.
Yeah,
with-
and you gotta reinvent. So I'm gonna run the blender. Hopefully it's not too.
Sorry, we could turn off the.
So I tend to run, I would probably end up running it a little bit longer than that. More surface area, more contact the microbes are gonna have with it. And also down the line, if we don't have a lot of stuff that hasn't, we're composting in this essentially. So things that aren't composted or been processed by the microbes are just down the line. Now there's stuff to get stuck into our hydroponic system. With hydroponics, it's a bigger concern. So I can pretty much eliminate, if I get this fine enough in here, I can eliminate most of the visible solids when I go to use it. And I'm going to go ahead and pour this right in to here. And normally I start up my air at this time. Yeah, I have a lot of solids. I didn't grind it as much as I normally do there.
Would you mind muting and unmuting? I think your audio just faded quite a bit.
Are we okay? Hold on. Okay.
Okay.
Maybe
How's
it's
it?
on
How
my
is
end.
it now?
I think that's better.
Okay.
Okay,
I'm
thank
not
you,
gonna
sir.
do any more of the blender. I think you guys get the idea. So I'll probably put like a couple gallons worth of this in. I would run it a lot longer in the Ninja normally, but obviously that's annoying here. Because I can see some solid as I go through, I can pick up solids in the system, and I really don't like it to have it this big. These are kind of like pencil head size. So I normally grind it a little further. That's okay. It's not the end of the world. It just helps
Here's the
later.
demo.
Yeah. Okay, so I'm gonna put in about a gallon or 2 of that. We'll imagine that. And then next I'm going to. It's gonna be messy. So this soil was taken. I like it because it's really active soil from other soils that I've had, generally in the woods, generally where people and animal, well, at least people's animals have not been. So like 1,000 foot from where known human traffic is. Not trafficking, just traffic, is where I like to pull it from. And then I go into the black spruce up here. I don't know if everybody has that availability in your area. I don't think you do. And I dig down to the ice lens, which is, or the top of the ice where I hit ice first. And that's generally within a foot in the black spruce here during the summer. And I take that soil that is just thawed right on top of the ice, and that's where I get this soil from. However, I have done tests with other soils here, and they are nearly equal. I mean, I don't know without using a microscope that I would see a lot of difference, but I see a lot of diversity in this soil, is what I like seeing. So I'm going to take that soil. Of course, it's going to have, I tried to get as much organic stuff out of it. All the, of course, fighting the black spruce is, we're right in the horizontal layer, the vertical root systems of that tree. So that makes it kind of difficult getting out. So I just try to get it worked down, and then I will crumble the soil as I put it in. And generally I put it in a container so I can measure it as I'm going. So that's what I'm going to do here. And I'm going to put about a quart of soil in with this, in with my 2 gallons of green material that I put in. And I talked about the different things I use. This is romaine lettuce that I'm putting in. I think chickweed brings a lot more to the table. I think clover brings a lot more to the table. One thing that I haven't experimented with was putting clover roots in with it. But there's this just stirs up a flurry of questions, especially students doing science fair, agri-science projects, to ask about these things. Doing 2 different teas side by side, making them different ways, seeing how the exact same plants under the same conditions react to 2 different teas. And of course, everything else being consistent. So I keep, usually it's wetter than this, but I keep turning that, getting as fine as I can. And then once I get that quart, I put it in. And at this time, I'm usually running my aeration, but it's really loud in here. And normally what we would see right now, the green material in there would already be off gassing and bubbling up in the cauldron, as I call it, in here. What we end up with after a couple weeks is a tea that's, I like to say it's about the same tint as a good southern tea, iced tea, when it comes out of there. In about 2 weeks is when I like to use it. And then it's free to use after that. And of course, if you're adding more to it, you might want to wait a few days before you use it again. Temperature. I find that it does work, is a little more active if the temperature is a little higher. I tend to run it here. These microbes, they're soil microbes, which of course our gut microbes are soil microbes as well. And of course, what do we run? 97, 98 degrees. And that's where they're the happiest. The ones that tend to happen in here, I usually run this around 65 to 70. And that's where I keep it. I have heated it before and tried that. It's not worth the energy to do for the difference. And the neat thing with students is you can constantly watch this. Okay, guys, let's pull a slide tomorrow. Let's pull a slide in 2 days, and then have the students report what they're seeing. And then if you do a lesson along with that on a quick identification chart of knowing different shapes, different motions, different structure of different kinds of microbes, while we may not be able to name the billions of different kinds of microbes that may be in there, we can see the families they're in. Are these good guys? Are these bad guys? And what form are they in? Are they active? We're only looking at active stuff. We're not looking stuff that's losing my words, in cells. What's that called? Yeah, when it's dormant, essentially. When they're dormant, we're not worried about them because that's good. So the bad guys should be dormant. They're there. They're ready to go if the conditions change. We're keeping it in an aerobic environment with the water going. So if our blower goes off in our water, we've got probably about 6 to 12 hours before we need to be seeing that going again, or we probably ought to dump and restart, because we don't want anything going anaerobic in it for these conditions. Obviously, we can compost in anaerobic conditions, but that's not this one. Full strength tea coming out of here, full strength. You can inoculate seeds with. It's arguable what happens. Some have argued that it removes the wax layer from the seed faster and it germinates faster. I don't know. You come up, make great experiment again for students to do. I had students take one to our state FFA convention on a study of different seeds that were inoculated with tea and their effects on them. And it definitely, we've seen seed germination within 8 hours using the tea on it. So that's pretty interesting. And of course, you have to, how do you define germination too? So, you know, is when the radical, you know, is present or whenever it is. Arguable again. So that's full strength. You can also use the, once you do the germination process, if you did that separately and treated them, you know, moisten the seeds with it and then put them to germinate in a different medium. I like to use rock wool. Mel's an advocate of the cubes, which
The
are
rooting,
the rooting cubes,
root riot.
root riot type product. I like using a certain size cube that's relative to the whether I'm going to be running in a tower. Her root riots work really well in the towers like the lettuce grow tower. And I like to use them in my deep water culture, and the little 1-inch tiny cubes work best for that. I have some in here. Yeah, so a smaller cube, they're 1 by 1 by 1 cubes. And it gets down, unit cost is down less than a nickel a piece. So that's why I really try to use the smallest thing. And just sticking with lettuce, this works really well. Larger varieties, we might want to consider a larger medium for. Or at least you would have to move them at some point. And when I go to the systems, when we go to our tower or when we go to the deep water culture, we dilute it, the tea then, and we go to a 1 to 16 approximately, 16 gallons of water to 1 gallon of tea from the bioreactor. Foliar, I haven't messed a lot along with, but it could be used in full strength or in a dilute as well, to address immediate concerns with your plants. I just haven't really explored that. And like I said, there's so many questions to ask. And what's really nice about the tea is it's safe. You're not going to overdose with it. No, it's not in, students can be around it. It's fine. As a matter of fact, we find that running this bioreactor and having plants in this room, we had a teacher from Delta Junction, real live person we can contact you with if you want, who suffers from migraines. And she was just coming in to talk to me. And she said within like 10 or 20 minutes of being in here talking to me that her headache went away. And she said that had never happened before with her migraines. You know, natural stuff. I guess that's where we find some of our best remedies. I can't tell you all the science behind that, but this does off gas. It'll give the room, if you do this in a classroom, it'll make it smell like a fresh forest floor is what it's going to smell like in the room.
It doesn't smell gross.
No, some students aren't used to fresh outdoor smells, so it's unique to them. But those who've been out, you can tell the students who've been out in nature and been hunting and all, they know this smell very well because everything is derived from the forest floor. Is where it comes. So essentially what we're doing, so that's what I tell students. I said, okay, look out the window, assuming you have a forest outside your window. And I said, who checked the pH in the woods today? Who did the ppms out there? And I'm like, what are you talking about? And I'm like, exactly. So in my system, I never check pH because my biology is doing my work for me in the ectorhizosphere of balancing that pH right in the root ball. And if it's checked there, you'll find it's right. It's ideal. It meets with the plant. The plant sending out its juices to get what it wants, puts its order out, just like driving up to a drive-up window at a fast food place. And if the proper biology is there to respond, we'll bring those things and mineralize what's needed for the plants. Pretty cool process. So because of that, we have less need for our salt-based fertilizers because now we're making better use of things. So yes, I do still use commercial fertilizers, but I'm only using 10 to 20% of what the lowest recommendation is on the package. So generally like a powdered fertilizer, I like to use, and Mel probably advertised these as well. I use the General Hydroponics green grow. I gotta remember what it's called.
You got
We have a thing of it here somewhere.
Captain something.
Oh yeah,
what is it?
Maxi Grow. Maxi Grow.
that's
General
it.
Hydroponics Maxi Grow is the product I use. And it recommends 1 to 2 teaspoons per gallon in a hydroponic system. I'm using a half a teaspoon, a quarter teaspoon in a gallon when I go in with it. And when I dose, when I make up water in the systems from fast evaporation, transpiration, I often will alternate. I'll put in fresh water. And then the next time I'll add more at that low rate back in it. So I keep that really low. That was a very difficult lesson as an old farm boy from the Midwest, that smaller might be better sometimes. Big concept to get over, because I always thought bigger was better, and I killed a lot of plants thinking bigger's better when I first started into hydroponics. So we've got it to, you can use the product right through harvest. It's not going to cause any issues. I wouldn't do any foliars of any kind near the end, but and that's also a nice thing is you don't have to wash your produce when it's coming out here. If we're just doing the veg cycle on whatever we're growing, for example, lettuce, it's, you can pick it right off, eat it. The plants are protected. Now we don't have to have a sanitized growing area where, oh no, don't go into the growing area. You'll contaminate it. You'll kill off all the crop because all the germs and things on you. No, we didn't have to wear protective gear when we went into the forest. And now we don't have to wear protective gear when we go into the hydroponics area. So now the toddlers can experiment with the plants and touch the plants and not have fear for them. And that's really it's so basic, it seems revolutionary. Because that's what we've come to is this, it's like we put plants on life support in conventional hydroponics. This actually makes life just so much easier. You can see, it doesn't matter if I put a little extra soil in there, or if I don't put a little extra, or if I put a little extra lettuce or not put a little extra lettuce. If it's 2 weeks or it's 13 days when I use it, probably not going to make a big difference. And that's really the gem of it all. Is it's so nice, it's teacher friendly. It gets us back to, it's a regenerative thought. It's, and I would even go as far as it's indigenous thinking, as far as how we're growing plants. Koreans have been doing this. I think you guys explored that at the down, and I'm still not all up to date on how Koreans use this method, but it's a similar mindset of creating our own. Mel likes to call them fertilizers. It is bringing something to the table, but the challenge is always hey Scott, let's take this and take it to a lab and analyze it just like we would standard fertilizer. Ain't gonna happen. It's not the same as fertilizer. It's not a synthetic fertilizer, therefore we can't analyze it.
But it makes the plants grow, and that's the most
It
important
does.
thing.
It does. It gets that communication going on with the root systems
They
and
seem happy.
gets it what it needs. And just with some plant material in there, seems to give it exact, a little bit of fertilizer. It doesn't quite, where it lacks is in nitrogen. You're not going to get, I've tried to grow plants just with this, I can't finish them out. I can't finish out lettuce with just the tea. It will start them, it'll grow mid-cycle, but after mid-cycle, it doesn't happen. You need a little bit of commercial fertilizer.
A little bit of boost.
And I've messed with that. That's why I started growing the clover to see if I could boost nitrogen in my tea. I don't know. I'm not
Eat something for you, Kelly.
a chemist or a
Good times.
microbiologist. Yeah, I'm not dangerous enough to know these things. So again, more experiments, things students can try to answer. That's the big question I have, but of course it's built in a lot of small experiments, is can we develop this, or better yet, we have 3 of these. This tea we start off with when the plant's in its first 2 weeks. This tea we use in its next cycle, and this one we use in its final. I wish it was more like corn, because corn is staged in how it grows. I staged the miniature romaine lettuce and I just don't have enough resources. I don't run a laboratory here. This is a modified storage room that we work out of, and I'm not always here. So it's very difficult for me to run any really conclusive investigations. But those are great things. I like to pass those on to students. It just gives the interested student, the motivated student will take off on this like wildfire. And like, oh, well, what if I did this? Well, what about that? And I learned this here in class. Well, how can I apply that in my study? What didn't I address?
I don't know. Do you guys have any questions? I think you covered it.
Yeah.
Does anybody have questions?
I got a few. I don't want to take too much time, but maybe I'll ask one and then let Max or Heather ask if they have any questions. So I guess my first one is having the water. What do you think about using rainwater? Is that if you can collect rainwater off a tin roof?
I would say yes, as long, yeah, I can't, it's gonna, even probably even have better pH, I would say.
Yeah.
Yeah, I would say yes, yes,
Okay,
yeah.
all right.
I wouldn't see why not, no. We have a pathogen in my water, my well water at my house. I live out in the contaminated flats next to Fort Wainwright, where they've dumped lots of great stuff out there. And for some reason, I have a pathogen. And what we do to water our plants at home is we just fill that. We use the same principle, but I put a table or like a teaspoon to a tablespoon in a 55-gallon drum of water and then blow it back down. And then we don't have any issues. So it kills off anything that's in it.
What do you
So-
put in it?
Bleach.
Oh, bleach.
Bleach, yeah,
Oh, okay.
yeah.
Interesting.
So yes, I think that's a great idea. Great.
Hey Max, are folks using rainwater out on Kodiak too, or what are they doing?
Mainly from the tap, city water. So just need to let it off gas.
Yeah.
That's been something one of the farmers, he's really adamant. He off gases all of his water. He braises it to room temperature to not shock the plants. The
Yeah.
others, they have their drip system on hooked
Right
up
off,
to the,
right off.
yes, and
Well,
it's
if you're
really
gonna have-
convenient. So
If you're going to be counting on your biology, off gassing is essential. Yeah, you're
Mm.
just gonna
Yeah.
cut into the population big time with a chlorine component.
How
exactly.
long for the off gas?
There's actually formulas out there written. So you could, if you have an open top container, not a necked container, there's a formula like how many ever gallons are in it. There's how many hours it'll take. I think like a 55-gallon drum takes like a day or 2 days without any air pushed into it. I think would be a safe number. And
But
then
if you've got air stones in it,
the
it
next
goes a
day,
little bit
next day I'm using that.
faster.
Yeah, and I use a highly scientific smell test. I really
And for like a
this
five-
system.
What was that, Kelly?
5-gallon bucket?
Yeah,
How about like for a
5-gallon bucket.
5-gallon?
With an air stone, with a basic air stone in it, overnight, it's ready to go. Yeah. Otherwise, I would give it a day or two just sitting there, and it'll still off gas. Yeah. So you don't have to use the energy. If energy's if we're teaching energy reduction, it will off gas by itself. Sealed, it won't. Yeah. I was going to say something about water.
So Max, just a quick question on the plants that those guys are growing. Do they off gas the water or before they use it, or are they using it straight out of the tap? This question. Out of curiosity.
Well, that's why it's a little bit of both. I think when I was talking about the one farm, he does that especially for the soil. So that's
Yeah.
just re-emphasizing that of why you're off gassing it. And I even off gas even just for my house plants. I always, it's a little thing you can do to like not kill your soil quicker. Very small thing.
I've noticed some of the schools that we work with that didn't do it, and they ended up with some serious chlorotic spots on their leaves. And then they were like, "Why has it got spots?" I'm like, "How long did you let? Did you use? Oh, we just used straight water." Well, that's probably what happened. So
Yeah, and I know for hydroponics they used to some of them, but then if you're using all conventional fertilizer salts, so you're learning that you really don't need to because there's not biology. But if you're trying to, build that biology, you got to get back in the habit of that.
yeah,
and all
that's
that.
true.
Yeah, and
I've worked
oh, sorry.
I was gonna work places where you're using the UV filter to kill the everything in the water. So you're kind of going the opposite route. And I started thinking, maybe instead of killing the water, maybe we should try and feed the water. And so I have algae, which is what you're trying to kill the UV filters, but we have hopefully bacteria and stuff in there too.
Yeah, algae is one of the first things that comes up with a new hydroponic grower. I think
Yeah.
that's always the first phone call we get is about algae. So algae is a minimum amount of algae is quite acceptable and expected.
We always say that.
So what we want to eliminate is like on towers is having sealed up so there's no leaks. As soon as it starts leaking, you get algae blooms. And if you're running a deep water culture system, is not having exposed water. And then the other,
But like,
yes.
Or is it just the light? You want to keep the light off the
Yeah,
water?
Yeah, keep the light off the water. Yeah, as much as you can. What was another one I was going to mention? And keeping water moving. If you want to bring you can bring water quality lessons into this, and moving water is healthy water. That it's being dissolved oxygen is being put into it constantly, and it's on the move. My deep water culture systems, I'll never run a one tub system. I always run 2 tub systems, and I circulate water between the 2 tubs. Heather Bogardus has these systems at her school as well that move the water between the 2 different ones. Plus, we introduce air in with air stones in each of the containers still. So always on the move, always introducing air where we can. Seems to reduce, but if the light gets that water, like if the mats aren't cut to the right size of your container, then you get exposure, and that tends to be where or if it's like an eddy essentially in your deep water culture, oh yeah, go gangbusters there. It'll look like a Midwest hog farm pond. It's yeah. There was another lesson. If you want to talk to students also about conventional agriculture and how this technology is being used in that, they've turned it, of course, they're monetizing it. So what they're doing is using technology to separate the different bacterias and fungi out, and they're making specific applications to crops, always foliar. They never improve the soil with their systems, but they're using biologicals, they call it. So they treat it, they end up spraying like corn, for example, 6 to 8 times throughout the season. And they're putting in biology that helps with nitrogen uptake, and then different needs of the plant at the different cycles of the growth of the corn. So from a soil health perspective, we're not getting very far. But it doesn't need to be. If a farmer brewed his own and applied it to the soil, all right, maybe even did an additional application in the season, his soil would be so much better, and his plants would thrive equally. But that's not how we do things in the big party.
Yeah.
Sadly. Yeah, I actually approached one of my friends in the Midwest about, I said, well, hey, why don't you do this? Oh, I say, that's too much work. And they're making better yields, and they're making just a little bit more money than they were not doing it. So that was enough to sell for them, and they don't have to worry about anything. So there you go.
Question on the details. How long are we supposed to brew it once we do our first batch before we use it?
2 weeks. 2 weeks is
Okay.
and again, there's probably some buffer either way there. Yeah, and it's really fun to watch in the microscope throughout those. The neatest time is about 2 days afterwards for kids to really look at it through a microscope, or anyone. Adults, it's like playing in a sandbox, watching stuff in the microscope. That's fun. And then having a basic ID, a basic microbiology resource to identify different things that are going on in it. And when the kids start seeing those, that's really cool, or we do. Yeah. And what we'll see is as the time goes on through those 2 weeks, you'll see a lot of decomposition, survival of the fittest, and you'll see a lot of protozoa fatten out. It's pretty wild to see. Yeah. Yeah, I actually have videos. They get down and watch protozoa munching away like Pac-Man. It's great. Yes, I'm sorry.
And how long can you hold it once you have brewed it? How long? So I have a 5-gallon bucket with it in there with the bubblers. How long can it just sit there and bubble?
I keep the air runs 24/7, and
Yep.
Mel told me we were going to do this, so I cleaned out my old batch, and I just had like 5 gallons in here. It had been running for months.
Okay.
Yeah, that batch had been going for months.
Just keep feeding it every now and again. You're good.
How
You
how
want
often
it
do you
to stay
recommend?
active. You
Okay.
gotta feed them. So
How often do you recommend feeding?
It's going to be based on, just like, think about mom's sourdough.
Yep.
It's
Yep.
soft as you pull away from it, and I like to refresh it when I'm not using it a lot. Just add a little bit of soil in it, and I'll add a little bit of feed to keep it happy. And you'll see it off gas again. You know it's working. And of course, if you're never sure, pull a slide and see what's going on. So I recommend a good shadowing microscope to look at your biology.
What power microscope?
I run mine. I can tell you.
I inherited
Look
some
at
old
it.
microscopes with
Is
my
that what that-
classroom, but I'm not sure they're strong enough to do this.
Bogardus, what is that? I think Liam's been able to take a good look with just your basic classroom
Dissecting scope.
microscopes there. What do you know what power those are? I think mine's a 400 that I use.
I don't, but I think ours are smaller.
Okay.
Even
But,
just
but I know Liam has been able to see, that things are going on. And when we looked at biology at a 3rd grade class, and they just brought
Yeah,
out
we
their
just
basic
use-
classroom ones, and they did fine.
Yeah,
I
we
guess-
just use the
Okay,
basic
cool.
dissecting scopes. Yeah,
I honestly think I'm not a guru on it, but I think a clean microscope is the microscope you want. Because
A clean
I've
one?
got one that's gotten dirty, and it doesn't look the same way it used to.
Okay.
dirty because it got water in it or what?
I move it around a lot. We've taken it to different classrooms, and it somehow is just accumulated dirt. Yeah. Oh, I was also going to bring up something. Oh, once in your system. So conventional wisdom is that we get salt buildup, and we need to change out our water on a regular basis. I ran a deep water culture system at a school, and we ran it the entire school year, and never changed it out, and never saw a reduction in rate of production of the lettuce.
Okay.
So that tells us we're not accumulating. So the amount of salt but wait a minute, Scott, we're putting salt in because we are still putting in the conventional fertilizer, the salt-based synthetics. If our
biology.
biology will consume that amount of salt. It's when we overpower that we're not consuming it. Yeah.
I heard Swiss chard's good too to pull salt.
It might
Oh,
be.
yeah. Oh, that's cool. Yeah.
I wonder if you used
Someone-
it as your.
Again, so many
Green.
questions, and
Oh, that's a good test, Kelly. You're on it.
yeah, and like
Yeah, I heard
Mel had
Swiss
me,
chard helps to reduce that. So
Cool.
and
Well,
they do not
It does
attract a
grow
lot
in hydroponics.
of insects, but Mel and I have been throwing in marigolds on the corners of our in the towers. We'll put in a larger variety of marigolds, and I'll run them on the corners of my deep water culture.
Well, and
yeah.
I have an interesting suggestion for you. I learned that this summer, Kelly. If you do have issues with aphids, try Nicotiana. It's a flower, and you can grow it in a hydroponic system. Nicotiana.
What? Say it again.
Nicotiana. And it has these little trumpet like flowers, and
Uh-huh.
the aphids are attracted to it, and the nicotine in it will kill them.
Oh, okay, cool. That's one of the nicotine family. Yeah, we used to use tobacco plants in our
Yeah, it's
row-
often called a
rows to try and-
tobacco flower is another name for it, but it's not tobacco, and it's just in that family. But that I was thinking about getting some seeds and trying it in our system because we have constant problems with aphids in our office. I was gonna go buy some from Johnny's and try to sprout them and put them in there and see if that would solve the problem. Because
That's a fun idea.
my friend was putting them in his greenhouse this summer, and he was like, "Oh yeah, it solved all the problems. We haven't had any issues with aphids since we
Awesome.
started putting the Nicotiana in there."
Nice.
So I'm
Yeah,
gonna test it.
cleared everything out for the summer and closed everything down, and that got rid of, I had white flies, aphids, and thrips, and we got rid of everything. So just give it 2 months with no food, and it's kind of sad tearing everything down. But I can't get in my building during the summer consistently, so it's just a good excuse to destroy everything. A lot of times farms can't destroy everything for 2 months every year, but I'm a farm school, you can. So that helped us. One other question. So you're saying our deep water culture back here, you can kind of see we're
Oh, yeah.
running two systems with about 140 gallons in each system. So you'd recommend about 15 gallons each?
For each one. So you have a basin down below?
Yeah.
Yeah.
Okay. So what?
So I have like a 40-gallon basin down below, and then about 100 gallons up top in the actual reservoir in the growing area.
I did those calculations early on. So what I've tried to do is Mel and I have worked a lot with teachers up here, and we're trying to make the most teacher-friendly system available. That was our main objective, teacher-friendly. And so that you can leave on the weekend, not worry about things, least amount of maintenance, care.
Mhm.
So what I did was eliminate the basin. So I put, and I found that snow sleds work the best. You probably saw those that we're
Yeah.
using. So now we eliminated a whole component from the system. And I probably run 4, 25, 50. I run 50 gallons in that system, and that's running 60 plants. 60 plants, and does that sound about the same ratio?
In each system, in each
Yeah,
plant.
there's 30 plants approximately in each one, and it's about 25 gallons. So it's about a gallon. Was that sound about the right ratio? Similar ratio?
Um,
Or are you doing it by
I didn't
plants?
catch the ratio there.
Yeah.
You said
Cause it's by water, how much water? Is that what you're asking? How much fertilizer to put in it? Is that what you're asking?
yeah, the mimetic onatai, the
Okay. Oh, yeah. So no matter how much water you have in your system, 16 to 1. Yeah. So if you're running
16 to
150
1.
gallons, you're putting in 10 gallons
Around
of
10 gallons of
yeah.
fertilizer.
ish gallons
Okay.
of the Medic one tie. Yeah. Yeah.
All right, cool.
And you can back off. That's the big mind change for me is just better be safe putting less in.
So speaking of being safe, we're doing aquaponics. We have a couple systems with fish in them. What do you think of adding this in with fish? Do you think that's okay? Or do you think the fish will keel
My
over?
quick initial thought is I tell the daring students who say, "Can I drink this?" And I'm like, "Yep, you may have to go to the bathroom a little sooner than I do, but sure."
Okay, it might kill the fish.
So
but it may not.
I don't think it's gonna. I think, and again, another great question. And I think my initial thought is yes, it should be safe.
Yeah,
Yeah.
Okay.
Well, that's a comment I was gonna make is that'd be a fun thing to try because
test it.
aquaponic systems are meant to break down organic, but like that's what they're made to do. And so it'd be cool to try a hydroponic system, but build it like an aquaponic system in terms of having a biological filter to that component.
We-
And then maybe you could
I like it.
Brilliant mind,
I
brilliant.
want to
mind.
know how this goes,
so,
Kelly.
Yeah, we actually talked about this process being involved in with it would be actually be part of the system. So the biggest argument I got from scientists as I talked to them about it was, well, that process may eat on your roots some.
okay.
That I've heard a lot of things that are true and tried them and had no problems at all. So
My only concern about with the fish is if you do, you got to just do it in gradually and then watch the ammonia levels, because if they're getting so much from the plant and the plant is not able to deal with the ammonia coming from the fish, then your ammonia levels might get really high, and then that would kill off your plant. So the plants will tell you.
Well, and that's the
Okay.
key. Don't use fish though. So you build
Yeah.
an aquaponic system, but the fish aren't actually a part of it. The system is just meant to break down biological waste in terms of
Yeah.
its design. Whatever biological waste you put in, that could be the experiment. You could put in whatever stuff, but it's
Yeah.
meant to actually like keep breaking it down maybe further than this processor. That's kind of something I'd be curious. Yeah.
Which is
Good
one,
question.
and it's not fresh on my mind. We're talking about incorporating a classroom scale biodigester in with this process too. So that's something I'm working on. I'm not going to talk about it yet because I am not well versed.
Yeah.
Yeah.
Yeah, that's
Well,
super
cool.
exciting.
Well, it's. Do you have any more?
Use-
I say have fun learning. I think this is a whole world at our scale, the classroom, smaller production, Alaska. I mean, we have so many elements that are different than anyone else has done. What Liam's discovered at North Pole High School is, and I tell him it's probably going to be different than what I discovered here in my office. So, or what variety of plant you're growing, in specific conditions in your place. His ventilation looks different than my ventilation. Yeah. And when COVID was on at the school I grow at, the plants reacted a lot different because they bumped up all the HVAC systems. And as soon as that was over, to save pennies and dimes and dollars, they cut back the HVAC systems, and my plants respond, humidity changed in the classrooms after
CO2.
COVID. Yeah.
Yeah, plants like that CO2.
They did.
Yep.
We did a little experiment. And one of the best ones we did different mixes of mimetic onatai with different concentrations of fertilizer, and having half fish tank water with mimetic onatai and half strength fertilizer did really good. So less like 1 EC at 1.0, but then using the fish tank water also seemed. So I'm thinking that kind of might be what I do is I pull fish tank water out of my fish tanks and put them in my deep water culture for the bacteria.
Ooh.
So I'm wondering if the fish tank back water mixed with the mimetic onatai and like lower, like half dose fertilizer might be
Oh,
Might be a good
yeah, we
one.
gotta write
There
this
you
all
go.
up and we'll
This-
put
this
it
is
on
great.
the web.
This is great.
I love it. This is
I've
great.
heard these questions, but I haven't heard anybody trying it.
Okay, I challenge you to get those kids to write this up and then we can put it up on the AFPC website.
It'd be great if they did a poster with it, like a science project poster. And then
Yeah.
that's easy to understand and communicate
I
with.
love it. I love it.
Yeah.
Ah, yay.
Are you guys, is King part of the FFA program now? Did you guys take on FFA?
No, because
Okay.
we have students coming from all the different schools. We don't have any, I have like 2 students in my program who are full-time King Tech students, so they have to
Okay.
bus off right when class gets out. And so
Okay,
I was told we just don't do after-school clubs or anything, and told no, but we have
Okay.
East is doing FFA, so we have one school in Anchorage doing FFA, and I might be able to look into like revisiting that conversation.
yeah. I was just gonna say it's a good structure. If you don't have science fair already, the agri-science is a science fair. And it just gives a structure for students. But you
Okay.
can use whatever, you can use ISEF, or you can use whatever structure you want, or the boroughs or the school systems structure for science fair.
Okay.
Yeah, have them go win a prize with it. That's great.
Well,
I pushed this all off, like Liam at North Pole. I'm like, he says, yeah, do you want credit? I said, nope, use your name. We're gonna introduce you, you're gonna present it. And that's why he was down there in Kodiak. That's exactly why.
You did a great job. Yeah.
All right, well, it's almost 3:30. But I wanted to thank you guys all for coming. And we have another one scheduled for next month, but I got no speaker yet. If anybody's got ideas, send them my way. I've got a thought of checking in a few places, but I felt if we have some more ideas or anybody has a topic they want to discuss further that we can just have a good discussion about, I think that would be pretty awesome. So let me know, and hope you guys all have a great month until we see you again.
Thanks for letting me share it today.
Thank
Thanks for
you.
taking the
For
time.
presenting. All
Great
right, everybody
presentation.
have a great day, and
See you later.
hope all is good. All right.
Take care.
Let's have
But-
You.
Heather,
I
you
wanted
stand
to-
on?
I just wanted to make sure that you saw the one in the corner.
Oh, nice. Oh, yeah.
Ah,
You got it all together finally. Yes, that's great.
Oh, did you get that piece?
We found one.
Yep.
We have a
It's-
bucket full of
We have the piece
odds and
like
ends
sitting
back
here.
there. So
Yeah, we're supposed to bring it to you.
This is
That's
supposed
okay.
to be in North Pole right now.
that's okay. We found the right size PVC pipe, and then I just went and bought one of
Okay,
the.
sorry about that.
No worries. You guys are busy. We're good.
Oh,
All right.
Heather, we also talked, we had a meeting today, a company meeting, and we were talking about educational opportunities here in the next school year. And we mentioned that you were interested in having us come out and teaching something natural resources or
Yes.
agriculture, however,
Yep.
and how we could best help you. So we're still interested in that, I know that you're overloaded.
Yeah.
So if you want to still, if we can help you and not overload you more, but take some weight off, let us know.
Absolutely. And if you guys ever need to I know some places have to get stuff like that on their books, and if you guys ever need to get some outreach done, I'll be your guinea pig. We'll come up with something.
We
I
do
teach
have a new
environmental
program.
science, so
It's called Roots, and it deals with all the stuff we do internally. It's stream bank restoration, invasives, hydroponics, everything that falls into that category. Everything we do here. And I'm doing workshops, offering workshops, but I need to do pilots. I need to go test them. And it
Awesome.
won't be new news to us, but it'll be a new presenting it. Where we've done hydroponics a lot, probably won't do a pilot program with that.
right.
And
Yeah.
but I'm going to do a stream bank restoration one, which soil or Fish and Game and Fish and Wildlife have always taught that, and I've sat back and watched, but I should be able to teach that one myself. So that's one I want to kick the tires with as well.
Yeah.
Awesome. So
All right.
do you have a copy of the school district schedule, like the bell schedules or anything?
I think I do. Well, I have Effie Cochran's that may be a
All right.
little bit modified.
I'll send you North Poles.
Okay.
And then my environmental science is 4th and 6th period. So on Thursdays, those two classes are alongside each other.
Okay.
There's no gap in the middle. So those are a great day to do a presentation on Thursdays, or we could do any day. And if you only want to do one class, we'll do one class. It doesn't matter.
We'll team up on that.
We can figure it out.
Yeah, we'll team up.
Awesome.
Mel and I will, and we'll put something together and let you know. And make sure it's the topic you want to.
Oh, twist my arm. Come down and see you.
Well, I mean, I think everything you guys do falls under environmental science.
Okay.
so.
Good, good, good.
Yeah.
All right.
Awesome. Thank you.
Later.
Bye.
Bye. All right.
Oh, did I?
It was still recording.